Happy Wednesday to you guys. I've been wanting to do this video for a little while, and I'm working on my YouTube channel, and I've got a lot of really cool genetic engineering experiments and stuff that I can't wait to share with you guys. But in the meantime, I thought, hey, let me just do a little instructional video or educational, whatever the hell you want to call this thing, and teach you guys what is DNA, what does the word itself mean, because when I began to study DNA and I actually looked at the word deoxyribonucleic acid, which is a mouthful for most of us, I realized there's actually a lot you could learn just by breaking down the parts of that word. So let's do that. Let's look at DNA. DNA. So I'm going to write out the different parts of the word, and then we'll go into each part and we'll just briefly talk about what that is. And I'm sorry in advance for the reflection, but okay. So we have DNA. Deoxyribonucleic acid. And I'm going to start with the easiest part of this word, deoxyribonucleic acid, and that is the acid part of it. Well, let's look at acid. Now, if you think back to your high school chemistry or biology class, what do you know about acids? What are acids? They typically have a pH less than 7. They also conduct electricity, and they typically have, or they do have, a negative charge. So they have a negative charge. And that's really interesting. I never thought that DNA can conduct electricity, and why does that matter? Why does it matter that DNA is an acid and it can conduct electricity? Well, there's something called electrophoresis, and it's this really cool way to visualize DNA. And it does that by drawing a current across a gel, and the negatively charged DNA will begin to create bands and move across the gel. Now it lets you visualize it's normally something DNA is so small you can't see, but with electrophoresis you can take advantage of the fact that the DNA is an acid and it has a negative charge, and you can use electrophoresis, which I'm not going to talk a whole lot about now, but you can use that to visualize and figure out the size of a segment of DNA, which I think is really, really cool. So that's the acid part. Pretty easy, I think. So let's jump into probably the most complicated part of it, and that's the deoxyribo part of it. Well, if we think about a strand of DNA, we probably all have seen something that looks like this double helix thing, and I'm not a very good drawer of DNA itself. There we go. You have these two strands, but at these joining intersection parts where the nucleotides meet the backbone of the DNA, which is called, another big word, phosphodiester backbone, but we won't get into that right now. One of the things that the phosphorus joins in each part of the segment of DNA is a sugar molecule called ribose. And the sugar molecule looks something like this, that you might have seen again from chemistry classes. You've got this carbon ring with an oxygen up here, but down over here in ribose, there's usually this hydroxyl group, an OH, an oxygen and a hydrogen atom hanging off of it. Well, what do you think the deoxy in the deoxyribo means? It means without the oxygen. In fact, it doesn't. In DNA, there's no oxygen here. It's just a hydrogen. And that's what the deoxyribo indicates. It indicates that obviously there's the sugar called ribose involved, and that this version of it does not have the oxygen down here, which is pretty cool. Now in RNA, there actually is RNA is a single strand of DNA that doesn't have the two backbones, it has one. And on that version of the sugar, it actually has the oxygen. But in DNA, there's no oxygen, and that's why it's a deoxyribo. Now let's talk about the nucleic acid. Nucleic sounds very similar to nuclear or nucleus, and it just really indicates that this is a biopolymer. It's a long chain of a molecule that typically lives in all living things, which is generally true. I wouldn't say it lives in the nucleus of all living things, because not all living cells have nucleuses. That's where the difference between eukaryote and prokaryote organisms, but that's sort of what it indicates. It's made up of a long chain of these sugars and nucleotides. It lives in pretty much every living thing. And so if we put all of that together, we have deoxyribonucleic acid. Deoxyribo because it's a long chain, it's a polymer, which is really just a long chain of molecules. It's made up of the sugar ribose. It's missing that oxygen. Nucleic acid, it's an acid. It conducts electricity and it has a negative charge. So there's a lot we can learn about DNA, and I just thought that I get excited about learning about this stuff. So I hope that you guys are having a wonderful Wednesday, and I can't wait to share more updates with you and to also see yours as well, and to hear from you. What are you interested in learning about DNA? What makes you excited about learning about genetic engineering? Okay guys, I'll talk to you soon.